All right, so for this lecture, I wanted to talk about pedigrees. Pedigrees are important, particularly in human genetics. It's one of the major tools that can be used. I know you've all sort of dealt with some pedigrees in the past, but what I wanted to do is maybe hopefully add a little bit, refresh some information, I should say, to start with, and then maybe add a little bit of information, show you examples of how it can be useful. So let's take a look at what we have. And here, let me change over to a pointer for a second. There we go. All right, so what I want to do is talk a little bit about human genetics relative to pedigrees, talk about standard symbols and the standard ways that pedigrees are set up, just sort of more a reminder there, plus maybe a few new points. And then standard series part 2 is there's a paper that I've included under the module sections where there has been a sort of major agreement on how some symbols are put together and what symbols might be used. Then we'll talk just very briefly about non-standard symbols. Those are becoming more important. Give a few examples, just fairly simple things. Then a very complicated one to show you how it's actually used in medical research and human genetics research to try to understand some disease structure. And then just a little bit to include in your notes that you can automate, of course, pedigree drawing. And there are both shareware, freeware, and pay for programs out there, depending on how complicated you want to make things. Okay, so let's just look at a pedigree for a moment. This is a classic pedigree. What you would see is that you would have generations. They're identified usually over on the left side with their numerals, so 1, 2, 3, 4, 5, 6, whatever generations you may have going back. Within each generation then, you have the individuals are generally numbered. And of course, there are different symbols for males and females. Squares are generally used for males. Females are circles. Then once you have a union of two individuals, a line's brought down, and you have all the progeny identified. And the progeny are identified as generation 2 in this case, in order, in birth order. Okay, so this first individual here is individual number 1, a male. So a son was born, was the first one. And they had a second son. Then they had 3 daughters, a daughter here, daughter here, and daughter here. So 3 daughters, so 2 sons, 3 daughters. So this was a family with 5 siblings, and they were 2 males and 3 females. And then you have matings, of course, next generation. So you have relationships between this first son and a female, and they have their children, and again, in birth order. So when this couple produced children, they had a daughter first and a son second. This couple with a son and daughter-in-law for these 2 individuals back here, produced a daughter first, then a son, and then a daughter. And then this one was son, son, daughter, daughter. Okay? And then they have 2 individuals which either have not reproduced at this time or not married. So they're just indicated as being progeny that exist. Okay. Now, there's lots of other things that are often put on here. Obviously, you've got darkened and lightened circles, darkened circles indicating some component of the phenotype that you're interested in, often a genetic disease or genetic problem, sometimes other things that are not necessarily genetic. Then very often you'll see the age of the individual put here, the birth age or the death age if they're passed away. There might be other information depending on what you're trying to collect. Remember, pedigrees, while we use them for simple modes of inheritance type analyses, they're also very useful for just other sociological and other types of data that are collected along with them. So, you may see all sorts of things, the marriage date here, the birth dates of these individuals themselves. These in some pedigrees, these are lengthened, so if this child is born first and then it's 2 years to this one, but it's 5 years to this one, this line actually may be lengthened out. It's just, there are a number of ways to do it, but in a standardized way, it pretty much looks like this, okay? So then the question becomes, what do all these symbols mean? What are related? Well, that's where we're gonna start with our first set of symbols here. And you have, as I said, females are generally circles, males are squares. Now let me say that In the past, there has been two primary systems. There's the European coding system for pedigree symbols, and there was the American system. And this paper is an attempt to pull all of that together under sort of an international norm. Unfortunately, it's still not the way it works, okay? If you go on the web in particular, there's just all sorts of things. You'll see carriers shown like this, where they're half and half. You'll see carriers shown with slashes. You'll see carriers, it just depends. Where you go, how you look. It's unfortunately never been standardized to the real degree. Now it is at the professional level. When you go to a professional society of genetic counselors or professional societies where they're looking at these types of pedigrees, they have a symbol system, okay, and you follow that system. But when book publishers and textbook publishers and others come out, they tend to put them in various ways that, whatever fits their mode and their system. So you'll see all sorts of things. But in general, individuals that are afflicted, okay, that you're interested in, particularly from a genetic standpoint here that we're talking about, are usually solid colored in. Carriers may or may not be colored in. If they are, they're usually half in some way. Deceased individuals usually have a line through them. Okay, so these deceased female and deceased male. If the gender's unknown or you don't care, you may go back a few generations and things and you don't really care. Ours is not known. If the sex of the individual is not known, then you just put it a little inverted triangle. Spontaneous abortions, miscarriages, are usually a symbol such as this or this. This is fairly common. This little dot here was incomplete. Okay, and there's other forms that you can show here. Now, you can shade these as well. Okay, if it was known to be related to whatever genetic disease problem you're looking for, you can color it in or bring it through. So you'll see those in different ways. Stillborn is usually, obviously stillborn is a non-thriving human, so it usually has SB underneath it and then a slash to indicate that they made it to term, they made it to parturition, but they weren't a living human. And then terminated pregnancies are those that usually are symbolized like this. So would be an abortion of some type, so a purposely terminated pregnancy. This one, the carrier of sex-linked, this is certainly a carrier of X-linked. It's the way it's used. Remember that you can also be sex-linked and be on the Y chromosome, which is fairly rare, but we don't have a lot of traits there. But most of our that we think of as sex-linked are like this. So this is a carrier of a sex-linked recessive. Okay, you don't see that one that often. It pops up in where you know that it's going on, but it's not one that's just in a standard. You won't see it in most textbooks. Well, here's a couple of them that cause a little bit of trouble. Dizygotic twins and monozygotic twins. These symbols seem to vary all over the place. Okay, this is a classic American version of what paternal twins are. Dizygotic means, of course, that there were two eggs ovulated. They became independently fertilized, but they went through the process together and were born same time. So they're fraternal twins. They're not from the same egg. Monozygotic, of course, twins are a single fertilized egg, which once it starts early division, breaks into two and goes on to become a complete set of individuals from a common fertilization event. What's different here is that this monozygotic twin standard is often looks like this, with this little triangle going back to one spot, going back to the top up here like this. Okay, when I said it wasn't always the same, see if I can get my pen to work. And here's not want to work. Come on, pen. There we go. When I said it doesn't work, sometimes what you see is you actually see this a dizygotic being here without this line drawn like there. Other times you'll see monozygotic twins without this little connection in here. So it looks, sometimes, you can confuse them with dizygotic. Most of the time, dizygotic is drawn where it's just simply straight back to the line like that, okay? And then monozygotic almost always has a drop down before you start to divide them. So there's something like this. So that part of it usually indicates a monozygotic process, whether or not you have the crossbars or not, okay? Just a slightly minor little problem that you see there. Okay. All right. Then, of course, you have mating, okay, going on. Consanguineous matings are usually 2 lines, meaning inbreeding. And very often it'll tell you right above the line whether it's first degree, second degree, third degree relatedness to individuals. Extramarital relationships that are important, okay, a lot of those, I mean, they don't report them if they're not important, but if they're important, if they produce offspring or something, then they will be indicated with a dashed line, okay? And then just as I said, first generation, second generation, birth order, and very often we ask you in genetics to put the genotypes underneath, okay? If you have an adopted individual, they're usually put in brackets with the dashed line indicating that they're not genetically related to these individuals, although they were part of the family unit, okay? And then the last of these symbols is what's known as a proband. Probands can either be shown as individuals who are afflicted, so there'd be afflicted probands, so that'd be someone who appeared to have a problem and they traced it back, or you can just have an individual that's not that as a proband. It's whoever started the genetic counselor or the investigation into the pedigree, all right? Well, these are pretty much the standard symbols. When you get to non-standard or more advanced symbols, you can look at some of those. Now, this paper, I said, talks about the pedigrees. And one of the first thing that's kind of important is, you know, what's going on? What are the instructions up here? What are you trying to do? Well, you're looking for the names of the individuals involved. You may want to try to get the ancestry, all these other things that you're trying to do. You provide some information. Now, here's what the kind of more accepted version of how symbols should look comes in this paper, and this is really taken from the paper that I've posted for you. First off is, you know, males, females, again, they can put the age at the time that the pedigree was done. They can talk about the birth date. They'll talk about a D for a death date for a deceased. They show you all sorts of things. Again, gender not specified. These are individuals who have it. And then, of course, as you'll see as we sort of close out this lecture, a lot of times you're dealing with multiple phenotypic outcomes, multiple phenotypic variants at one time. And you're trying to figure out, okay, this is someone who's got this, you know, particular genetic disease or genetic problem, but they also have a series of other things as part of that syndrome. So if you want to illustrate a number of different things, you can do it by splitting them up either into halves or fourths or something else. And we'll show you how that works. If you're just building a large pedigree and you don't, you know, it's way off from the third cousins and you don't know anything about that side of the family or whatever, but somebody tells you, oh, Aunt Margaret had 5 boys, okay, then this is really how that's symbolized. Okay. If you don't know how many individuals there were, say, you know, go back 2-3 generations, say, I don't know, my great-grandparents, I don't know how many brothers and sisters they had. Okay. Well, you can just put some form as unknown and this. Okay. Again, deceased individuals are lines drawn through, and again, you often put the death at what age. And that can be important for things where you've got diseases that are early onset and trying to understand it. Again, these little arrows are the proband arrows. Consultant means that that's an individual who just came in for consulting. Maybe it was a fertility clinic, maybe it's something else. And then probands are individuals who actually had a disease themselves. Now again, this is not standard. Even though it says it's standard here, it's standard to see the little arrow indicating it, but the little P right here, this proband thing isn't always present or not, okay? But if you see a little arrow pointing toward an afflicted individual, you know it's the proband, okay? Then we have things like, again, stillborn, usually information about when that occurred. And if someone is pregnant when you're doing the pedigree, again, pedigrees are, you know, they're in real time. There's information about it, and maybe you know that there's been an issue. This individual here would be a male who's got an extra 21st chromosome. So they apparently are there was a test done and they were determined to be afflicted with trisomy 21 or Down syndrome potential. Okay. Again, spontaneous abortions, miscarriages, information that you can indicate about that, pregnancy terminations, etc. Epitypic pregnancies are highlighted as being that. Okay. Then we have some other symbols that fit in. So this is how we would put things all together, right? You've got your basic lineage here, okay? A break in the line, maybe there's been an early family and then a divorce, and this is the current family that you're working with. Maybe the proband's off over here somewhere. Usually just a break in the line one way or the other indicates that this male had a previous marriage, previous relationship with someone here. Again, consanguineous is multiple lines. Biological parents that you're dealing with are here. Under these rules, this is what, a monozygotic is supposed to come down like this with the 2 lines, branching off later. Dizygotic comes in like this, indicating a single birth with 2 different progeny. Again, these could be the same, all right, because fraternal twins can either be both male, both female, or 1 male, 1 female. And you also put the birth order here. If it's a fraternal and it's a female is first, a daughter, you would have a little circle here. Then if it, a few seconds later, minutes later, you have the second birth, you put it here. So birth order still plays into here. And again, a lot of times this line right here is not there. You actually goes just up to the line up here. You'll see that. And you, of course, you have trizygotic and everything. Okay. Family history not known, just question marks. No children by choice or something. It's just an end line right here. Just to know it might be a tubal ligation or a mastectomy that's played. If there's infertility, and a lot of times it's used in infertility clinics. Oh yeah, my cousin, they were never able to conceive. They tried and things. And if you know the reason, and there are lots of different reasons if known, there. Okay. And then of course we said earlier, brackets are for adoptions and how those adoptions occur. Okay, and this is just an adoption from outside the family here. Okay, this is adoption from where you adopted, not uncommon in many situations. If maybe a loss of a parent over here, that you would formally adopt your nephew or niece or something like that. So forms. Okay, then of course it gets more complicated, right? In today's world, we have lots of different types of relationships. We've always had different types of relationships, but in a genetic sense, we have the capability to move sperm, to move ova, to eggs, fertilize individuals, to have outside of the couple births. We have all sorts of arrangements that can occur. So there are representations for that. D represents the egg or sperm donor. That's the donor. S represents the surrogate. Okay, you got that. And you can have all sorts of other combinations. All right. This is sperm donor in this case, right? In this case, you would have maybe there was azygotic sperm here or something. There was a problem. They're not able to reproduce, so they go to a fertility clinic and they use, select sperm from some donor. Then that's brought into the system, so you know that this individual is not directly related to both these parents, but is half related here. Okay. Again, this one's showing that they tried and they were unsuccessful reproduction forms. This is an oval donor, of course, female right here. So she provided egg, and you can see all the different ways. Here's a complete surrogate individuals over here. In this case, the sperm was taken from the male, eggs were taken from the female, but maybe there was a uterine problem or something, so he could go to full term. So they used a complete surrogate female for this child up here, right? And again, this one is going to be genetically related to these two, and it just as any other child would be. It just simply was brought forth here. These, of course, are not, because you've got donor situations, and then you've got all sorts of other combinations. I'll let you work through those. And these are planned adoptions, where you go in and you actually negotiate an adoption or something. A reason for that is that, if you're tracing genetic issues and this is the proband, or this is part of the process of the procedure, you kind of need to know that this individual was not the product of a mating between these two, right? That's not where it arose. It arose outside of that, therefore it may not play a role, or it may be very important that, and particularly if you have any information about these donors, you may want to include those. Okay. Right. Well, those are, again, you can look through that paper. It's very good about outlining some of how it is and why they've gone with some of these standards, and you may see them used. Okay. Now, a couple other things we'll talk about. One is the non-standard pedigree symbols. We have, as I said, not necessarily collecting just simple information about whether it's a dominant trait or recessive trait, and whether there's an affliction. A lot of times with these pedigrees, we're using them for other social pieces of information to help us understand behaviors, understand other things. So what you may see, they may see that there's a sexual orientation notation put in. One of those is that you can have, of course, a gay in the male, and that male would be symbolized as a male square. A female would be symbolized as her circle. Then you have a modification of that, which then in some case maybe a little triangle in the middle to indicate that this is a lesbian relationship or two females or something, or males. That, you know, and that can be important because you say, okay, we've got this male, and that male has used their sperm with a donor to produce a child, right? Even though it's a gay couple, whatever. So you're just trying to keep, you know, so you understand the flow from generation to generation, and the individual's direct relationship of where the chromosomes and your proband came from. And then there's, you know, other symbols like bisexual male, bisexual females. You know, it seems like that to me that these gender identifications and sexual preference identifications we're in a time when they're changing at a very rapid rate. We have pansexual and all sorts of things, and that's fine. It's it will standardize at some point, and from a genetics standpoint, sometimes that makes a bit of difference. Sometimes it makes no difference at all. But you know, there's been some attempt at least to capture some of that information in fairly symbolic ways when it's necessary. Okay. Ones that are, you know, alternatively very necessary to understand too, or when you look at individuals, is whether they've got a transgender individual, particularly if they've made transitions, okay? Not just whether they're declaring themselves as a transgender, but whether they've actually started or completed the process. So, you know, if you have a male here that has transitioned to a female, this is one of the preferred symbols. In a female that's transitioned to a male, and this would be a symbol, or these are very common symbols, particularly these bottom two are when there's transitioning going on. It's not complete. This is a male transitioning to a female, the female transitioning to a male. So I just put these up here because you know, there's certainly much more awareness of these situations in today's society. And so if you're working in medical fields or things, you may likely see these types of things somewhere on a pedigree and go, oh, I wonder what that is. Well, now you should know. Okay. Well, why don't we look at just a couple of these and then play with a couple and see how you do. Let's go back to some very simple things, right? Autosomal recessive, if you're doing a pedigree, you know, the big key to that of autosomal recessive is that neither parent will have it, and that some, you know, progeny of outcome of theirs will have it, right? So that would be autosomal recessive, was at least. Now, when you're doing these simple little pedigrees and problems that you might do in a book or somewhere else, always use Occam's razor, right? Start out with the simplest explanation you can find, okay? Could this be an X-linked, you know, X-linked recessive? Well, sure, because female, she could have given the egg, the X chromosome in the egg to the son. Neither daughters would have it, so you got the classic crisscross pattern and all that. So could that be? Absolutely. Is that the simplest explanation? Absolutely not. Okay. The simplest explanation is go all the way back to sort of Mendelian, and then work your way up to the more complicated ones. Autosomal dominant, there's always a single rule for that. Somebody's got to have it. Some parent has to express it, right? If it's a dominant, could be both parents, but one parent has to express it. Okay, you can't have a pattern like this first one over here and have a dominant, right? Unless it's some hidden, we're not getting into that. But on a basic pedigree, you've got to have it. So in this one parent happens to be a female, and she's producing progeny here. Okay. Now, alternatively, of course, you look for, you can have sex-linked, and that would be sex-linked dominant, sex-linked recessive. Or it can be Y-linked, Holandric. Holandric's the easy one, right? When you think how you know to look in a pedigree for X-linked or sex-linked or these, is if you have a big enough pedigree and you look for it only to occur in one gender or the other, okay? First, is it only in males or is it only in females, okay? And then, okay, it's not that, it's in mostly males and most, you know, but every now and then there's a female. Well, it still can be X-linked, okay? You just look for the preponderance. You expect, because of just classic, you know, think about it, XX and XY matings, You expect a 50-50 sex ratio in the progeny. Okay? About half the kids are going to be male and half are going to be female. So, you expect that's what you would see here. If you see these pedigrees, long pedigrees, where, yeah, you're still getting the right sex ratio, but the afflicted problem, the thing you're after is going down male groupings, or it's going down female groupings, or they're majority males or majority females, then you may potentially have a sex-linked issue. So X-linked, right? Crisscross, daughters, dads send it to daughters. Okay, this is X-linked dominant. He's got to have it. So dominant, okay? And all the daughters are going to get it. Why? Because all daughters get an X from their dad, right? Sons, they don't, right? Sons get an X from mom, and then they get the Y from dad, so they don't carry it. X-linked recessive, a little more difficult, okay? X-linked recessive, dads don't have to have it, okay? But what you will see is it's crisscrossing, right? It will crisscross over from mom to sons because sons only get one X. Where do they get that X from mom? Now, if she's a carrier like this, she's a heterozygote, only about half the sons are expected to express it, but none of the daughters are expected to have it, right? Now, you can have an X-linked recessive on the males, right? So like this male right here is to mate. You would expect, expect none of his sons to have it, okay? And it would depend on what the female was involved, whether the females would have it, because it would be a recessive, right? But certainly he's not going to be able to pass it on to his sons. Now, what he can pass on to his sons, or when you have a holandric condition, like over here, carried on the Y chromosome, that's going to just directly pass down lineage after lineage after lineage through the males. That's what I said. So just look to see if you got it. All these males, all these females, or some disproportionate combination. Like here, the daughters all had it, but the son didn't. Okay. All right. So let's just try a couple and see how we do. What would you maybe think this looked like here? Okay. Well, what are the rules? Well, it's not a dominant, right? Because the parents don't seem to have it, but some of the kids do. These parents don't have it. Some of the kids do. And if you're doing a really big pedigrees, one of the things you often end up doing is making a hypothesis about it being scientific, making a hypothesis about it based on some part, like over here, and then you trace it around to other parts and see if that holds. And in some place, if it's not correct, it'll fall apart usually. Okay, big pedigrees, that's really how you do them. You pick a smaller part of it and try it, okay? Well, what do we got here? We've got males, okay, have it. Okay, is it sex-linked? Well, there's a male that doesn't have it, and things. So that's not the easiest explanation. By far, the easiest explanation here is probably just an autosomal recessive, right? Here's a small group. Now, of course, you don't, let's pretend these are hets and hets, right? Heterozygotes times heterozygotes. So neither one express it, but they have a Mendelian probability of a 3-to-1 progeny. Well, they just didn't have enough progeny, remember, when we talk about those 3-to-1 Mendelian ratios, we're talking about producing thousands of fruit flies or hundreds of corn, parts of corn in a year or something. Humans, you're not going to see those ratios because there's not enough progeny produced. Okay? But is that consistent? If I say it's autosomal recessive based on this, is that consistent with this grouping? Absolutely, right? Neither have it. He was probably a het. She had to be a het. They could push it down. So that's the simplest Occam's razor explanation. So what about this one? Well, we've got two males. We've got a stillborn right here. Okay, don't know what that was. Or spontaneous abortion, I should say. Sorry. This female had it. Both sons get it. And then this individual has it, and this son gets it. Okay, so it goes straight down through here. Everybody gets something. They had it. It's probably dominant. Okay, that would suggest that it's dominant because everybody that gets it expresses it. It's probably not a sex-linked dominant. Okay, that would make sense if we just looked at this part of it right here, because you'd say, okay, she's giving out X's. Okay, and that X could be, carrying whatever, and the sons are getting it. Okay, it's also, if we're going to go that rule, it could be an X-linked recessive. She could be a colorblind female. So both X's, both of them have the colorblindness problem. Her sons are all going to be colorblind. Okay? So that could be that. Well, how about, what does that say over here? Well, that doesn't really work, right? Because dad's not giving any X's to his sons. So it's not something on the X's, it's got to be somewhere else. So I think if you look at part of the pedigree, you see multiple possibilities. If you go somewhere else, it'll rule out some of those generally. So I think probably just a simple autosomal dominant, right? And then what about this thing? Okay. Well, we've got all kinds of little stories here. We've got this thing going down here. So it looks like it might be a dominant. Okay. And then we've got this not showing it here. So, but it's a crisscross pattern, right? A male's giving it to daughters. That's part of the sex linkage sort of more motif. And it's not going here. We don't know much because we only got one progeny over here. So, it could be an autosomal dominant, right? Because she's got to be recessive. If it is a homozygous recessive, that means all the progeny are going to be, or autosomes are going to be heterozygous. And so she's just not passing it on, right? Is there another recessive? She's just not passing it on. He doesn't have them to pass on, so you don't see it. Okay. Autosomal dominant. Could it be an X-linked dominant? Daughters. Yeah, absolutely, right? She could be a het. And for the X's, and just send the non-afflicted and non-dominant one down. Same story, right? It could be an X-linked. Okay. Or could it be an X-linked recessive? Let's think. Okay, it could be X-linked recessive. Well, he's definitely got it and expressing it as like color blindness. She could be a carrier for color blindness, right? She could be normal X and then an X with a colorblind mutation. The daughters could get it for sure. Okay, and then would this daughter pass it on? Not unless the male's colorblind. He's not going to pass it on to a daughter. So if she'd married a colorblind male, yes. So it could be any of those, right? So the problem with these tiny little pedigrees is, I don't know what this is really. Okay, I'm going to say, well, you could, Occam's razor says it's autosomal dominant until you get further data. This is one where you'd want more and more information. So just point out that some are pretty straightforward, and you can rule out things. Other times it's not quite as easy. Okay. All right, well, let's look at something that's a little more complicated. All right, this is the real world. Okay, this is a series of pedigrees. Now, this is not just one pedigree, but this is 1, 2, 3, 4, 5, 6, 7 different family groups, occurrences of certain things where they're trying to figure out mostly what Kallman syndrome is, okay? And they're seeing things like delayed puberty, okay? That's this symbol right here. Remember I said if you got multiple things going on, so this had delayed puberty. This is someone who's died who had delayed puberty, but their children, at least one was normal, one had a male with delayed puberty, and you've got all sorts of other things going on, okay? Now what we're interested in is this individual that has Kallman syndrome, and that's this individual right here, number 4. Okay, that's this female right here. That's this male right here. Okay, and it's but you've got all sorts of other things. You've got cleft palates. You've got all sorts of operations going on here. Okay, here's the consanguineous mating, where one of the children has this. Here's the one where again, whole lineage back here, a bunch of you go back many generations, but they're just dead. They weren't able to tell you a lot about it. And again, here's where you get these really weird things going on where you're having to jump around and jump over generations and then here's relationships that are outside. I mean, it's a very, very complicated pedigree, but what we're looking for are things like these individuals here, these individuals here, okay? And why am I showing this? Because it's just a complicated pedigree, okay? So if you look at that, parts of it, you might say, well, that could be a recessive, that could be something. It popped up here, and neither one of them had the full-blown thing. They apparently had delayed puberty, but they didn't have all this. There's all sorts of things that it could be. So what is Kallman syndrome? Well, it's hypogonadotrophic hypogonadism. So hypo means not enough, and it's when you don't produce enough hormones out of the pituitary to have full maturation of primary and secondary sex characteristics. Okay, and it's a weird system because most of the neural parts of your brain originate near or at those areas. Okay, So if you've got you know, a lobe of the brain, the cells that make up that lobe originally originated near there, okay, and they didn't move. But it turns out that some of the ones that are found in the hypothalamus that are very important for secretion of these sex hormones actually start out in other places. And one of those starts out in the nasal plecode, and then migrates over through a channel during fetal development. Obviously, is what we're talking about here, into the hypothalamus. Once it's in the hypothalamus, then it triggers the hypothalamus to secrete a number of different proteins and variations of those proteins that affect the pituitary. Once the pituitary gets those, it has receptors. The pituitary then responds and sends out steroid hormones that control things like completion of ovarian structure, dissension of the testes, adult hair patterns, pubic hair patterns, all sorts of things. There's this pedigree back here, Hunter syndrome, is when this doesn't happen. It's when individuals that basically never reach sexual maturity, even though they're living humans in their forms, they simply they don't descend the testes, they don't produce sperm, they don't ovulate. They're individuals who generally, you know, are not going to be individuals that. However, if you look at the pedigree, I say that. However, here's an individual right here who has the syndrome, who's had 2 normal children. Okay, and here, you know, others where you see, you know, this is basically somebody who has the syndrome plus other things, who isn't producing. Okay, they actually line probably has 2 measures here. You see other things, and then you see individuals here. So even though they have delayed puberty, they can, some can't, right? So trying to go back to this pedigree and figure out what's going on, because you have so much variation in expression things. Now, one of the things that they do have, and particularly in the strong ones, is that they have no sense of smell, or very poor sense of smell. The reason for that is that you have this dysfunction of this movement out of the development of these nasal plecodes. Okay, but as you see from the pattern here, they don't produce the luteinizing hormones. They don't get any feedback. They don't then have descending testes or sperm count's low. They don't get all the secondary things that you would get. Okay, muscle tension, ancillary hair, etc. What's going on here? Well, it is one form of it. If we go back, is a sex-linked recessive. Okay. And it's passed down from moms to sons here, okay, like this. It's an excellent classic X-linked recessive, and sons end up with it as cases for it. Other indications are that it's autosomal. Some of these you look at and you get, well, wait a minute, I got a daughter here and dad didn't have it, you know, so how did the daughter get it? It turns out that it has a force. Well, it turns out like many of these things, the reason I was pointing this out is that pedigrees, while they can be really interesting and useful, have limitations, right? Here's one where there's at least 25 different genes involved, because you've got to have genes that are going to develop these particular neural cells. The neural cells have to mature themselves. The neural cells have to migrate, be along pathways that will allow them to physically migrate within the neural tissue. They have to be in the hippocampus. There have to be receptors that are produced, as well as appropriate proteins. Some of these proteins are simply transcripts that are modified, edited. Okay, editing has to work properly. All that has to work in order for the pituitary then to put out the right hormones and the right quantities of those hormones. So there's a million steps in here that have to occur, and therefore there's a million, but there's many different genes that have to be involved in order for that to work. Okay, so just trying to put that, even with very complicated, very elaborate pedigrees that show the individual showing up multiple times and all sorts of things, you still have most genetic diseases have really complicated arrangements, even though pedigrees can be important. I mean, they can really lead you to an understanding, because in many cases, a majority of the cases that you can find in the world, it is an X-linked recessive. More males are going to get it than females, okay, just like color blindness in its forms. Okay. just wanted to show you something to give you, pull some human into this, okay, a little bit, and not just pedigrees. And then the last little thing just to tell you quickly is, of course, drawing pedigree things is pretty, you can do it in PowerPoint or on your Mac or whatever. You can make little circles and squares and connect them. It's not that bad. But there are pedigree drawing programs. Professionals use a number of different ones. There's some that are commercial. There's some that are free and online. I just wanted to throw this in there and make sure you had it in your notes. If you even want to try this sometime, I mean, you might try it for your dogs. It doesn't have to be humans or horses. But if you needed to draw a pedigree, there are ways to throw the information in there and get this. You don't have to sit there and try to make little loopy lines to do all sorts of things yourself. Okay. All right. Well, that ends what we wanted to talk about as far as these pedigrees. I think they're in the lecture, and we'll move on to some more information on the next series of lectures.